Hello there. This presentation will go through the steps to take for running the Denome human or mouse qPCR kit using a 96-well qPCR plate. Each kit comes with 34 pre-mixed primers, DT1 to DT34, provided in a 96-well plate. Before you begin, prepare aliquots of the internal control so as to minimize freeze-thaw cycles. You should also check your cDNA quality using the cDNA QC primer provided. CQ values obtained from the qPCR run should range between 16 to 19. To create a master mix plate, add 168 microliters of 2X SSO Advanced Universal Cyber Green Supermix into each primer well. Gently remove the 96-well plate from the box. Briefly spin down the plate. Carefully remove the strip caps from the plate. Hold the plate down and be careful not to spill the contents of the plate. Add 168 microliters of 2X Cyber Green Master Mix into each primer well. Gently pipette up and down to mix. Keep the plate on ice if you are running reactions on the same day, or keep the plate at -20 degrees Celsius for storage. Pipette up and down to mix well. Don't forget to change your pipette tips when going on to the next column. Keep your plate on ice if you're running reactions on the same day. If not, store your master mix plate at -20 degrees Celsius. To prepare your qPCR reaction plate, first label a new qPCR plate for easy referencing. On a new qPCR plate, label your wells 1 to 34. It is highly recommended to run 2 technical replicates per primer. Each 96-well plate can run a single sample with 34 primers and duplicate. (340 microliters of diluted cDNA is required to run all 34 primers in duplicate. However, it is recommended to prepare the diluted cDNA in excess and to store the remainder for further validation of target genes. Add 5 microliters of diluted cDNA sample into each of the 68 wells. Vortex and spin down your diluted cDNA before aliquoting into the qPCR plate. Add 5 microliters of diluted cDNA into each well. Using a multi-channel pipette, transfer 7 microliters of master mix from each well of your master mix plate to the corresponding wells on your sample plate. For example, from column 1 of your master mix plate, aspirate 7 microliters of master mix and dispense into column 1 of your sample plate. Aspirate another 7 microliters of master mix from column 1 of your master mix plate and dispense into column 2 of your sample plate. Remove the master mix plate from ice. Using a multi-channel pipette, transfer 7 microliters of master mix to the respective columns of your sample plate. On your sample plate, each well now contains 7 microliters of master mix contains cyber green and the qPCR primer, and 5 microliters of your diluted cDNA to make up a 12 microliters reaction. When running qPCR of Denome core genes for the first time, it is highly recommended to also run the internal control on the same qPCR reaction plate. Prepare the internal control reaction mix accordingly, and add them into the last wells of your qPCR reaction plate. Spin down your plate and seal with an optically clear sealing film. Your plate is now ready to be run in a qPCR machine according to these cycling conditions. Once the run is complete, retrieve CQ values and upload them to the Denome cloud server. Further instructions on how to upload your raw data can be found at our website. Do not hesitate to contact us if you have any questions. Thank you.